Thanks very much. My name is Ben Lawrence. I'm a medical oncologist from Auckland City Hospital. I also work as part of a translational neuroendocrine tumour genomics team, which is based at Auckland, but also with collaborators around the country. And I'm going to show some data today that's been collected by this large team, with especially mentioning my co-PIs, Professors Chris Print and Mike Finlay. One of the things about this title that Michael kind of gave me is that integrating cancer genomics and imaging requires that you understand both things before you put them together. And I think what's clear from Michael's introduction is that you understand a lot more about cancer imaging than we understand about cancer genomics. The other thing is that we, just as the same, when you talk about radionuclides and emitters and linkers and chelators and things, I often nod and smile, but I don't always know anything about what you're talking about. And there is some risk that if I talk to you about genomics that you may feel a little bit the same way. So hopefully I can find a happy medium and we can talk together collaboratively. So I've taken a slightly different emphasis, which is because we don't really understand the genomics very well, the implications of what we're finding in the new, cool, cutting-edge genomic understanding of NETs and what that means for NET imaging. Welcome, if you're from overseas, down south to the top of the world. And you'll also be pleased to see that you have also arrived right in the middle. And I guess one of the things down here is that we do things, take a slightly different perspective, and although we have the same conundrums, maybe some of the conclusions are a little bit different. So for me as an oncologist and you as nuclear physicians, what happens when you see a neuroendocrine tumour that's metastatic and it shows no avidity? Here, this is actually a solid lesion, you can see on cross-sectional imaging, which doesn't light up on a GAT8 PET. There's some avidity and FDG in the rim of some of these lesions. Why is that? One of the tendencies I find with nuclear physicians is they tend to think about what the physics is, a technical problem, something to do with the thing that you're inserting and trying to view. And what I want to encourage you to do today in the next 15, 20 minutes is actually take away the idea that some of that variation is from the biology, not the physics. Why do different cases look so different? And here's two small intestinal NETs patients of mine with their gallium PET. Really quite different distribution, different avidity, and different lesions show up in different ways. And why is that? But within cases, we know that there's enormous variation. So here's one of my patients with a bronchial neuroendocrine tumour who has had a great response to PRRT in the liver, but it did nothing initially to the bone metastasis in the lumbar vertebrae. And why is that? Here's a picture from one of Michael's papers that I've adapted with a GAT8 positive lesion in the top of the liver, not FDG avid, but a large FDG avid lesion which is barely GAT8 avid. Why is that? And what I want to suggest to you is that the answer is biological, and at least part of the reason for that is genomic. So the network program is something that we created in New Zealand about 3 or 4 years ago and have been building. We're trying to turn the whole country, which has about 4.5 million people, into one big neuroendocrine translational research clinic. So we have the patient's experience, which we record in a registry, so we have about 3,000 patients in this now, clinically annotated. We collect their cancers either from archives or fresh frozen if they're undergoing surgery or resection, biopsy. We're building our clinical experience. We have a weekly MDM, and clinicians from any hospital in the country can dial in on a video link to have discussion about complex cases. And we conduct deep genomics. And our vision is that anyone diagnosed with a neuroendocrine tumour in New Zealand first gets a genomic analysis of their tumour before we make the clinical decision. Be that PRRT, which we don't actually have funding for, sunitinib or everolimus, which we don't actually have funding for, or temozolomide, which we do, which is our only option since it's cheap. Here's our New Zealand, faced the other way up so you can recognise it, but this is 6 cancer centres, a spine down the middle of the country, clinical leads at each side, our lab group on the left, our Unicorn Foundation, our fantastic patient advocates, a key partner, great cultural advice, data managers across the country, ethicists down the south, our scientific advisory committee from around the world, our pathologists doing the heavy lifting in Auckland, our surgeons and interventional radiologists collecting the tissue, our sequences down in Dunedin, bioinformaticians around the country, and our incidental findings committee. So there's a lot of moving parts just to get to get each of this information, each of these bits of information for each tumour. So I'm going to show you the first genomic information that's coming out of this, the first 61 patients that we've sequenced with pancreatic NETs. Now, as you know, neuroendocrine tumours are really heterogeneous because they come from different places, they originate from different cells. What happens if you look inside one really tightly, well-defined clinical scenario? So, pancreatic NETs, well differentiated, resectable at diagnosis. Sounds pretty homogeneous, right? Well, let's look and see what's inside those tumours and try and take away some lessons for imaging. So this is a chart which shows genes down this side, different patient cases along here, and a coloured box is a gene mutation. And before we look at those, I just want to show you this panel on the right here. So the first thing we found with, to get patients into this study, they had to have a pancreatic neuroendocrine tumour that's well differentiated. So the pathologist who'd seen the case originally called it a pancreatic NET. Our two neuroendocrine MDM pathologists looked at it again. Yep, definitely a pancreatic NET. Matched up with ENETs and WHO criteria. Always, pathology is more subjective than us non-pathologists like to think, but so there's always a bit of discussion. But what we were able to do once we had some genomic understanding of the tumours was go back to them in 4 cases and say, actually, this isn't a NET. This is not a pancreatic NET anyway. This case turned out to be a pancreatoblastoma, and we could point to that because of the P53 and the APC mutation. These 2 cases here ended up being pseudopapillary tumours or solitary pseudopapillary tumours, whatever you call them in your institution, because we found a missense mutation in beta-catenin, which is pathognomonic in these 2 cases. And sure enough, when we went back to the pathologist, they said, well, actually, we had argued about that, and we thought they looked like a SPTT, but they showed all the neuroendocrine markers, and so we went with NET. And finally, this case, which I'll come back to, was actually a metastasis from a small intestine. So the first lesson from the genomics, if we extrapolate that to imaging, is that if it doesn't show up on your scan, one of the things to think about, is it actually a NET? And the genomics is helping us with that. It's helping the pathologists to say, actually, some of the things we previously thought were NETs aren't. So when your sensitivity drops, first thing, think about the biology. It might not be what you think it is. So coming back to these mutations, the thing to notice here is that there's a lot of MEN1 mutations, about half of the tumours. There's some other things we already knew about, ATRX, P10, and DAX. But after that, it's like as if you take a sampling of the mutations that occur in other cancer types and you scatter them across the pancreatic neuroendocrine tumours. And what you get, and those are the second, third mutation in the tumours, and what you get then is a random mix of genomic profiles inside each of these tumours. And all of them are completely different, with the exception of a few that have no mutations we could detect, but the rest of them are entirely individual. And so just from the very first principles, it's highly likely they look quite different from each other on imaging. So clinically homogeneous NETs, this is only one type, well-defined pancreas NETs, are genomically heterogeneous. The biology is going to be different, and it stands to reason that so is the imaging characteristics. So I'm only going to show you sort of 3 of these charts, and hopefully you bear with me. This is tumours along this bottom, and this is now chromosomes up the side. A blue box means that one of the 2 copies of that chromosome has been lost. And this is a staggering finding in neuroendocrine tumours. So you can see here that this, for example, this means this one here, these are all a blue box with a 1 in it, means that you've only got 1 of the 2 copies you started with left. So remember, a chromosome has on average about 1,000 genes on it, and one copy of that is completely gone. It's amazing that a cell can even survive like that, but nonetheless they can. So what does that mean for, if we take one direct example for imaging? I'll just tell you one other thing is that, just out of interest, not really related to imaging, is that these guys that have lost chromosome 11 here, this is chromosome 11, which is where MEN1 lives, that's one of the hits on MEN1, and the other is the mutation. So one of the key drivers of these pancreatic NETs is the loss of the 2 copies of the MEN1 gene, one by the whole chromosome going, and the other copy being lost by mutation. In the tumours that we have to worry about, that you have to worry about as nuclear physicians, you can see this yellow bar means metastasis, and you can see that those can occur in lots of different genomes. And if I just focus your attention on this box down here, staggeringly, these tumours, about 11 of our tumours have lost one copy of the same 10 chromosomes. 10 chromosomes, 1,000 genes a chromosome, one copy of that left. Who knows what that does to a tumour, but we have to treat them, and you have to treat them. So just what could this mean? Just a really simple example. So somatostatin receptors, 2, 3, and 5 are the most involved with gallium-68 imaging. And of course, DOTA-TOC and TATE, I hope I'm telling you the right thing here, tend to have more affinity for somatostatin 2 and 5, DOTA-NOC for all 3. But these all live on different chromosomes. So the gene that encodes the protein that's expressed on the outside of the cancer cell that you're trying to image, the target, is actually found located in different chromosomes. And if we go back to our aneuploidy chart and we think, well, what's there? So somatostatin receptor 5, and a lot of these tumours that we have to treat have lost half of the gene, half of the copies of the gene. And so it follows that, and often follows, that there's less of the protein expressed as well. So if you're trying to image a tumour which has lost the gene, one of the copies of the genes of the protein, it's highly likely that it's not going to be expressed as well. What about somatostatin receptor 3 on chromosome 22? Same deal. You can see a lot of these tumours that we need to treat have lost one of the copies. And you would expect that these tumours may not bind the receptor as well, and these ones over here that have a normal number of copies will be okay. Fortunately, somatostatin receptor 2, which is common to all of the imaging modalities, is usually, this dot means that you've got 2 copies and it's just a normal copy, usually expressed. But there are some limitations. There's a couple of cases here where there's only 1 copy of the gene that are metastatic, and we may have limitations in our ability to image those lesions with that genotype. Interestingly, the patients that have had PRRT in this sample, that means that they got through the test. They had avidity on a gallium-68 DOTA-TATE PET and therapy. They all actually have euploid genotype, which at least for chromosome 17, where somatostatin receptor 2 is located. So the lesson from this is that you better check that the gene for your target is actually still present in the tumour when you're trying to hit it. And so there's potential to work together in terms of understanding the genome of the tumour as you image it. So this is the next thing you need to think about is nets change when they spread. As they metastasize, the genotype changes. This triangle here is the liver, and this is the pancreas. And if we take one example here, this is an ATRX mutation in this pancreas primary, and also the patient has lost a copy of chromosome 3 and chromosome 11. When the tumour metastasizes, it changes. This one maintained the same mutation, but it only had loss of 3, not loss of 11. Now, this might not directly change your therapy, but you're starting to get an idea that it's highly possible that there are changes in the genome that change the biology and may well change your ability to see the spots. In this case, 1 subclone is spread to 2 locations. What about this case? This one has a MEN1 mutation that has loss of 5 chromosomes, including, interestingly, chromosome 22, which is where somatostatin receptor 5 gene is located. When this metastasized to lymph nodes, it actually picked up 2 new mutations to make that switch into metastatic lesion, but it did keep the all 5 loss of those chromosomes, or did still lose them, and therefore probably not change its imaging characteristics. And in this case, 2 new clone that occurred with 2 new mutations. So expecting that the biology will be different, and therefore the imaging characteristics are also possibly different. In this case, a neuroendocrine tumour was resected from the pancreas. It had no mutations. There was a loss of 3 chromosomes, 6, 18, and 11. A liver metastasis resected, an isolated metastasis resected in the same surgery, which had a loss of part of the same loss, but not the whole thing, so a subclone. And through somatostatin receptor imaging, a gallium PET, which became available in New Zealand shortly after that, they found a primary in the small intestine. There was concern that this patient might have 2 primaries, and the endocrinologist who had seen him was hunting around for a germline change But actually, we're able to see by looking at gene variations outside the coding regions that there are some that these tumours are all the same. They have the same signature underneath. And what had happened, this started in the small intestine, metastasised to the lymph nodes with one clone with a loss of 11, another clone went to the pancreas with loss of 3 different genes, and a subclone of that went to the liver. So the story here is that the imaging helped us to understand the genomics, but also the genomics is telling us that it's highly likely the imaging characteristics are going to be quite different of these tumours in the different locations. So NETs change as they metastasize. You may see different imaging characteristics based on that biology rather than a change in the physics. And finally, not quite imaging, it's more PRRT, but are there some tumours where we think where chemo-sensitising with agents such as temozolomide could be more useful? And just to cut to the chase, if we put on MGMT expression along the top, MGMT, as you will know, is an enzyme that's associated with maintaining genomic stability, and it's also a biomarker for sensitivity to temozolomide. And if we look again at that group of highly aneuploid tumours, you can see that green here means really low expression of that gene, and that protein that's encoded by that gene is required to repair the damage caused by alkylators such as temozolomide. So maybe, when we think about all these metastatic tumours with these yellow bars, it's these green ones, these tumours here that are massively aneuploid, that are the ones that are going to benefit from PRRT with temozolomide chemo-sensitising agent the most. So the value of chemo-sensitisation might well be predictable, and it might not apply to all of the tumours. So in conclusion, when we're thinking about reasons for no avidity, I have to ask, is it a net? It might not be, and the genomics can help us with that. Is the target gene for your target protein you're trying to hit still present? When you're seeing inter-case variability, well, we're seeing that all nets are genomically completely different, and we would expect them to be different just on first principles from the genomics understanding of biology. When we're seeing intra-case variability, well, we know that nets change as they metastasize, and I haven't even talked about the response to therapy and development of resistance. And again, this can be somewhat explained by the genomic changes in the tumour. Therefore, genomic variability in clinically homogeneous samples is likely to be reflected in imaging characteristics. Integrating genomics with imaging changes the way we see nets. To acknowledge all of my collaborators, and thank you very much for your time.
Well, thank you very much. Very exciting, and the talk is open for questions.
Looks like I slid a little too genomic, I'm sorry.
Yeah, please, here. Okay. Is there a microphone around?
There's a debate going on in when to treat patients with PRRT. I come from the Rotterdam group, and we're dealing with PRRT for more than 20 years now. One group says when the tumor is not progressive, don't treat it, just leave it. And try to treat when it starts growing and spreading out. But listening to you talk, might we not be better off in sampling the genomics of a small indolent tumor and base our strategy upon that?
Thanks for raising that. I think that's a great point. I was going to go backwards and show you a slide, but in the MEN1, just to answer by way of answering that question, in the MEN1 mutant tumours there are 2 clusters. There's the cluster I showed you that had that massive aneuploidy, and there's a cluster next to it. The cluster next to it, as far as we can see, don't relapse and progress incredibly slowly. And if you are able to sample the genome of the tumour, and you find that the tumour, that they're in that group where they just have a loss of chromosome 11, they don't have that massive aneuploidy, MEN1 mutation, they are going to behave well. And I would probably suggest that, we obviously need to prospectively test this, that's a group you don't need to treat up front. If you found that the genomics showed they're in the other group with the massive aneuploidy and the MEN1 mutation, I would suggest we need to treat those up front because they're going to behave badly. So you could make the decision, and coupled with the genomics, you could make the decision of whether to watch and wait or go straight in with PRRT assisted by the genomics. I think that's what you're asking.
Thank you.
Thank you.
A question. Have you any data on epigenetic changes in these tumors?
So the question is, are there any epigenetic change in these tumours? So we've conducted methylation, whole genome methylation analysis on 15 of the 61 tumours. And it's a hard thing to check, but there's no doubt about it that it makes a difference. So when we're looking at the pathway activation, for example, mTOR pathway activation, classically thought to happen by P10 mutations in these tumours, we actually find that the epigenetic switches that can turn on and off the gene expression are more dominant in some of these tumours than standard, what you would expect from the loss of a tumour suppressor such as P10. So, for example, there's one tumour where P10 is lost. P10 is a tumour suppressor on the mTOR pathway, so you would think that that pathway would be far more active. But actually what we find is that mTOR itself, the bottom of that pathway, is the promoter is hypomethylated, so there's no handbrake on that tumour. And MEN1, sorry, actually I'm telling you this story back to front. So in that tumour, mTOR is lost. There's a gene mutation in mTOR, and there's a loss of heterozygosity of the other allele. And so we're seeing that the DNA is not telling us the whole story. In another tumour where there's normal P10, you would expect that tumour to be sensitive, not sensitive to mTOR inhibitor, but we see a loss of methylation on the mTOR. That tumour is very high mTOR expression, and we would pick an mTOR inhibitor based on that. So methylation is definitely a player in these tumours, and I haven't shown much data on that. It's a harder thing to show, and I actually have to say that even though we're using Illumina, the Illumina chips, which are easier to analyse, they're still pretty hard to analyse, and I think that any groups at the moment who tell you otherwise are probably being optimistic. We're learning about it, definitely involved. And I think even more so in the small intestinal nets where there's even fewer mutations than there are in the pancreatic nets. Sorry, I didn't answer that very clearly.
Professor X.
Lovely talk, Ben. Thanks very much. You've emphasised the heterogeneity that exists at a genomic level. One of your former mentors, Irvin Modlin, has a test called the NET test, is looking at gene transcripts. And I sort of wonder myself how you take a heterogeneous group of cells that are all shedding transcripts into the blood from a whole lot of different tumours, and you come up with the kind of accuracy for telling you what treatment you need, what imaging you should have, etc. Do you have a feeling from that, from your experience with both that test and your own work with sampling specific areas of tumour?
Yeah, I have some worries about that test as well. I think that, if you're trying to sample, I wonder actually if imaging, some of the solutions that you guys are coming up with provide a bit more information in that in terms of expression of the somatostatin receptor indicating differentiation and FDG indicating aggressiveness. And I think that these things, there's a lot of heterogeneity. I think that the development of that particular NET test was based on statistical comparison as well as then some retrospectively fitted proliferative markers. But I think it's really hard to separate out the contribution of the white blood cells in that. And so I think it's going to be hard to make that test work. But, if it's prospectively assessed, that's really the only way of knowing if it's going to make a difference.
There's one more question.
Yes, hi. I'm Adiola, medical oncologist. Just a quick question about patients who relapse following PRRT. Do you biopsy them just to check for any genomic changes that could help predict, those who relapse, and also just as a way to actually gather information on genetic upregulation or downregulation on treatment?
The answer is at the moment we don't, unless we see a lesion that appears to be changing grade, in which case we do. However, in the future, we think that serial biopsies, or at least probably biopsies actually, of the most dangerous lesion is going to be required to make this type of approach work. I think that we know that tumours change when they metastasise. We know they're all different from each other, and we know that they change in response to treatment in their genome. And if we're actually going to find the genome, match it to a drug that switches off that exact gene change, then we need to know what the current gene change is, and that's a dynamic state. And so some people are trying to get around that by using circulating tumour DNA. That's a possibility that we're also looking at. It would be great if we could do that, but at the moment I think gold standard would be rebiopsy in the setting of a clinical trial, of course.
Well, if there's no further question, then thank you very much for this great talk.